Well, good morning, good afternoon, good evening, good night maybe for some people in the Far East. We are here in this lightning session number 1 of today, and it's about functional and structural components of tropical forest ecosystems, and part 3, so there were 2 other ones before. My name is Frans Bongers. I'm from the University of Wageningen in the Netherlands, and I'm very happy to be your moderator today. We have a fantastic lineup of 11 presentations, so that's quite a large number, of which many of them are early career scientists and PhD candidates, and so this is going to be exciting and new, and some people I've seen before. Megan, yes, I've seen you 2 days before. Some other people maybe also, I'm not too sure. There is a possibility for everybody to ask questions. You know the system by now, I guess, all of you. In the question and answer within Zoom, not within WOVA, but within Zoom. Meanwhile, I will look at it. You can, I think the presenters, the speakers can also write things in the chat. And please, if the camera starts with the presentations, that's about an hour, 11. So that's about an hour. Turn off your camera and turn off your microphone. That makes it the picture more quiet. I think that is it. I will now name all 11 presenters. So we have Elliot Morant from CATIA in Costa Rica. We have Megan Sullivan from Select in the USA. We have Chow Pham, University of Minnesota, USA. Amakantan Tirukamaran, University of Jaffna, Sri Lanka. Isabel Losa from Missouri in Puerto Rico. Marcel Carita Vaz from the University of California, USA again. Tan Pham from the Forest Institute in Vietnam. Yanar Dwikatyo, I think, Flora and Fauna International in Indonesia. Kalpana Madhushani from Sri Lanka. Sean Pang, University of Singapore, great. Sergio Oliveira Jr. from University of Sao Paulo in Brazil. Well, that's the lineup today. I look forward to it. This is going to be exciting. So please, I think that's it for now. So Javi, you can start the video. See you back.
Low-intensity selective logging does not negatively impact forest structure or diversity in an African tropical forest. Tropical forests globally are increasingly under threat due to profit-generating land uses. Selective logging is a compromise between forest use and protection. It generates income, but it does not completely clear forests from the land. Across the forest landscape, canopy cover largely remains intact after selective logging occurs. However, selective logging can result in changes that impact forest structure and diversity. How strong these impacts are and how long they last are unclear, but much of the variation seems to be due to logging intensity. We conducted our study on how low-intensity selective logging changes forest structure and diversity, and came up with these hypotheses. We expected that a temporary increase in the heterogeneity of understory light levels would occur after selective logging, and that negligible effects on basal area would also occur after selective logging due to the sparse nature of low-intensity logging. We also expected increases in understory tree diversity and density due to increases in forest heterogeneity. And we expected to find increases in understory liana levels due to environmental changes in selectively logged forests that benefit lianas. We conducted our study in Gabon, which is a country in Central Africa, and we set up vegetation plots across different ages of log forests in one logging concession in Gabon. We set up 80 plots in total and set up our plots across 4 different blocks representing 3 different treatments. Our 1 year since logging treatment is represented by the green triangles on the map. The 10 years since logging is shown by the red circles on the map, and we have 2 control treatments represented by the blue crosses on the map. Our vegetation plots look like this, and we measured seedlings, saplings, and adults in the plots and subplots across all of our different treatment areas. In total, we measured 10,125 trees and 1,394 lianas. We found that recently logged forests have slightly more open canopies, so higher mean canopy openness, and more variation in canopy openness, so higher standard deviation of canopy openness in the 1 year since log forests than in 10 years or control forests. We also found that saplings had higher rarefied richness in the 10 years since logged plots, and that saplings had higher species evenness in the 10 years since logged plots than in control plots. Similarly, we found that seedlings had higher evenness in the 10 years since logged plots as well. Sapling-sized lianas made up a higher proportion of the community in older log forests. So we found that the relative liana abundances in the 10 years since logged plots were higher than in 1 year since logged plots or in control or unlocked plots. In conclusion, we found that recently logged forests had a more heterogeneous light environment. We found that logging had negligible effects on basal area, so no real differences. We saw that there were modest positive effects on sapling diversity. Otherwise, selective logging didn't much affect seedling diversity and did not at all affect adult diversity. And the only evidence of negative effects that we found were an increase in lianas after logging in the older log forests, indicating that this may be the most effective area where management can improve by using liana removal techniques to deter lianas from growing after logging. I would like to thank all of the organizations and people that offered me support throughout this project, and thank you all for listening to this talk. If you'd like to contact me, my contact information is below. Thank you.
I'm Chofa, and I would present on how diversity, dominance, and topography drive above-ground biomass in the mixed dipterocarp forests of Sri Lanka. The relationship between plant diversity and above-ground biomass is highly debated. A lot of times, higher diversity leads to higher biomass, and this is due to a mechanism called niche complementarity. What this means is that species differ in resource and habitat use, and so the more species a community has, the more efficiently it can take up all the available resources and use them toward building more biomass. But the diversity of biomass relationship can also be non-positive, and this is due to mass ratio effects or the effect of dominant species. Some species may have certain traits that are unique or important that allow them to be very productive and very competitive. So a low diversity community dominated by these very productive species may actually have higher biomass than a species rich or species even community. And mass ratio effects is usually measured by the community weighted mean of some functional traits. In this example, they found that above-ground biomass increased with CWM of maximum height, but the relationship with species richness was much weaker. So here it is mass ratio effects rather than niche complementarity that is driving above-ground biomass. Another factor that can affect biomass is topography. A lot of times, biomass decreases with elevation due to the effects of colder temperatures on higher elevations. Biomass can also decrease with slope, since plants on steeper slopes are more vulnerable to wind throw and landslide. So with these factors in mind, we came up with these hypotheses. We predicted that above-ground biomass would increase with species diversity and functional diversity, indicating niche complementarity. Alternatively, it might increase with community-weighted mean of some functional traits, indicating mass ratio effects. And finally, above-ground biomass may decrease with elevation and slope. We have 24.5 hectare plots on an elevation gradient ranging between 395 meters to 815 meters. We used a mixed effects model with a random effect plot to test our hypotheses. Our predictors include species diversity, functional dominance, and topography. For functional traits, we used wood density and maximum stature, since these traits represent different life history and space usage strategies in forest trees, and they have also been used a lot in diversity biomass studies. Our response variable is above-ground biomass, measured in calculated in tons per hectare. So these are our results, and as you can see, there were no effects of species diversity, neither rarefied richness nor species evenness. For functional diversity, we found a positive effect of functional richness in stature, which may indicate niche complementarity. At the same time, we saw a much stronger positive effect of CWM in wood density, indicating mass ratio effects. So these two mechanisms, mass ratio effects and niche complementarity, are not necessarily mutually exclusive, and here they are both operating but on two different traits. And in fact, diversity and dominance with respect to these two different traits might have canceled each other out, so that there were no effects of species diversity. We found a slightly negative effect of slope, but a positive effect of elevation, which is opposite to what is often observed. But it must be noted that our elevation gradient is quite small. Our highest site is only at 815 meters, so there might have been negligible effect of reduced temperature on decreasing biomass. And instead, we found that this effect may have been due to the distribution of certain taxa across the elevation gradient. Or our collaborators, field technicians, and support staff who have been involved in the data collection process, and we thank the Yale Tropical Resources Institute for providing us with the funding for this project.
Hello everybody, good day. I am Thirukumaran, master student from Sri Lanka. I am happy to present my research work on floristic diversity and carbon stock estimate of a novel community forest ecosystem in Sri Lanka. Areas where for natural forest has been cleared often regenerated as novel ecosystem. This often because local communities enrich forest by planting preferred species and managing this forest collectively known as community forest. The study was aimed to access the floristic diversity and the carbon stock in the forest reserves. Next, we look about the methods and materials. Full enumeration was done in the forest reserve with a division of square plot at the dimension of 20 into 20 meter. The total of 1.16 hectare with 29 plots were demarcated by using GPS coordinates in the study area. For the assessment of floristic diversity, Sun and Vina index, species richness and evenness were used. Similarly, for the species dominance, the important value index was calculated. Pantropical allometric equation were used for tree carbon stock estimation. The population density of trees, saplings, and seedlings were 495, 2,307, and 5,160 stems per hectare. Sun and Vina index of trees, saplings, and seedlings were 2.19, 1.19, and 1.36 respectively. And this result shows that trees diversity was higher than sapling and seedling. Similarly, the evenness of the above categories was greater than 0.5, and this indicates the trees, saplings, and seedlings had less uniform distribution. Based on the important value index, the dominant species in the community forest was Mimosops elangi, followed by the Sadavakai and Garcinia spicata. The mean value of the tree carbon was 218.8 megacarbon per hectare, and it was contributed substantially by the dominant species in the forest reserve. The species area curve was represented the species richness had a significant strong correlation over the area. Tree height was increased at a decreasing rate with an increasing diameter. There was a significant strong positive correlation between the carbon stock and tree diameter of the basal area. Conclusion. Altogether, 44 species, including trees, saplings, seedlings, and 4 lianas were recorded from 29 families. Population density of the trees, saplings, and seedlings in the community forest was 495, 2007, and 5,160 stems per hectare. Dominant species are there. The mean value of the tree carbon stock, basal area diameter, and height were 0.43 megacarbon, 0.13 meter square, 25.49 centimeters, and 8.13 meter respectively. Way forward to develop the community in Sri Lanka, inverse species should be removed. It is recommended to conserve the germplasm and taken the necessary steps for multiplication of plants. Making the fence around the periphery of the forest and regular monitoring is recommended for the suitable community forest management. Thank you.
Thank you for being here. I'm Isabel Loza. Most of my research is focused on tropical forest, and I'm very interested on the study of rare plant species. In 2013, Ter Stege and collaborators published this paper about the hyperdominance of species in the Amazon. The most striking result was that only 227 species make up 50% of individuals in the Amazon, so most of the species are rare in terms of abundance. Then I became fascinated about why species are rare and how they could escape extinction risk. The study of rarity is important because it's related to extinction risk. Rarity can be measured in terms of geographic range size and local abundance, and also habitat breadth. Those are going to be called the axes of rarity for this presentation. Geographic range size is the area occupied by a species, so small geographic ranges are likely to be affected by adverse conditions. If we imagine that an event of adverse condition occurs in Costa Rica, then Ardisia pleurobotria will be more vulnerable to extinction risk than Cyathea caracasana, because the latter has a wider geographic range size. Local abundance is also correlated to extinction risk. Local abundance is the number of individuals of a species in an area. Small populations are more vulnerable to demographic and environmental stochasticity than species that have high abundance, which is the case here for Dendropanax jungacensis that has low abundance. And finally, habitat breadth is also related to extinction risk. Habitat breadth is the number of habitats that species occupy. Species occupying a small number of habitats are most vulnerable to environmental change, which is the case for Cereus jungacensis that is only found in one habitat in the Madidi region. We tested three hypotheses. The first hypothesis is proposed by Rabinovich, 1999, which states that there is no relationship between the three axes of rarity. So a species can be rare in different ways. Hypothesis number 2 is proposed by Brom, 1984, which states that there is a positive relationship between the axes of rarity, which means that if a species is rare in one axis, it's going to also be rare in the other two. And hypothesis number 3, proposed by Lesica and collaborators in 2006, states that there is a negative correlation between geographic range size and local abundance, a positive correlation between geographic range size and habitat breadth, and a negative correlation between local abundance and habitat breadth. We tested this hypothesis using data from the Madidi region that is in La Paz, Bolivia. We have there 48 large tree plots in a gradient of 200 to 4,000 meters sea level, and we have around 1,000 plant species, and for each of those species we measured geographic range size, local abundance, and habitat breadth. I'm going to show you the results now. These are the histograms that show the variation of each of the axes of rarity. As you can see, many species in the Madidi region have small geographic range sizes, low abundance, and are present in few habitats. This pattern has been reported for other regions and other organisms before. We found a negative correlation between local abundance and geographic range size, and also we found positive correlationships between geographic range size and habitat breadth, and between local abundance and habitat breadth. These results mean that we don't have support for any of the hypotheses proposed, as you can see in this comparative table. These results suggest that species with narrow geographic range sizes can compensate for this type of rarity with high local abundance, or a more uniform distribution of abundance within their range. It seems that this pattern is found especially in regional species assemblages characterized by a high proportion of narrowly distributed species, such as the Madidi region of Bolivia. And with that, I want to thank to all the people that make this possible. Thank you so much.
Hello everyone, my name is Marcel, and today I will talk about how the decoupling between adult and sapling niches may help tree species to coexist in this particularly diverse forest. So, tropical forest hyperdiversity is one of science's oldest standing mysteries. This is especially true for plants, who all use the exact same resources, light being the most limiting of them in these forests. Therefore, the most important coexistence mechanisms must involve the partitioning of light gradients. As you can see in this figure, this is a three-dimensional gradient, but let's first focus on its vertical component. Could the vertical light gradient provide enough niches for so many species of trees? To test this idea, we worked in what is possibly the world's most diverse forest in the central Amazon, where we have this large permanent plot that harbors hundreds of tree species. We then compiled adult height data from the literature and herbaria specimens for most of these species, and found an amazingly high number of species sharing the same adult niches, especially below the forest canopy, as it can be seen in this table. Seeing this quite uneven distribution of species across strata, strata, we realized that focusing on within strata coexistence would be more important than understanding vertical segregation per se. Theoretically, species sharing the same adult niches could have contrasting regeneration niches, which in turn can be inferred from sapling traits. For example, this is a short sapling with a large crown who maximizes light interception at the cost of growing tall. That is, it has a shade-tolerant growth strategy. Conversely, this tall sapling with a reduced crown is employing what we call a shade-avoiding strategy. Based on these assumptions, we sampled hundreds of saplings and measured 3 architectural traits: height, crown projected area, and crown thickness, and 2 covariates: stem diameter and crown illumination. Analyzing this data with Bayesian statistics allowed us to describe early growth strategies for each one of those sampled species. When comparing the dissimilarities in adult and sapling traits, we found that although there is a connection between them, which is indicated by this empty corner here, saplings can still vary a lot within any given strata. Now, because adult and sapling traits are largely decoupled, within strata coexistence could be possible if species sharing adult niches are spatially segregated. This segregation could be caused, for example, by environmental filtering of sapling traits, like is shown in this diagram to the right. Here we see 2 understory and 2 canopy species regenerating in different patches of the forest. Note how in this case, competition within strata is minimized compared to competition between strata. What we found, however, goes against this hypothesis of spatial coexistence for not only sapling traits, but also adult heights were spatially autocorrelated, as can be seen in these maps. Interestingly, these patches are not the same between these 2 maps. So light might not be the only factor creating these patches. So although the mystery of within strata coexistence continues, and by extension, the mystery of this forest's hyperdiversity, future attempts to map the forest's 3D structure, like this one here, that was done with LiDAR technology, will certainly help us move forward and unveil this mystery. Actually, a friend of mine has already started doing this mapping in the same forest where I study, so wait for the next chapters in this story. Thank you so much for watching, and many thanks to all these great supporters. Have a good one.
Hi, my name is Tân Phạm, and I am from Vietnam. I am going to talk about the research that I did as a part of my master's degree, which characterized the butterfly diversity in Central Highland, Vietnam. Like most places, butterfly of Vietnam is affected by latitude and other factors of biogeography, and Central Highland is where several endemic and rare species were found in the last 20 years. It has been fully studied by the fact that it has an unique flora and climate, high elevation, large and extensive forests. I address the question, where are the butterflies you find at high elevation, typically has narrow species distribution. I also examine what environmental factors that determine what individual species can be found within a landscape. Applying a standard method, pollen work for sampling butterfly, I worked at 4 transects. I also compare my own data collected in a couple of decades to have a broader picture of diversity and spatial environmental parameters. 8 butterflies that we found, we classify them by on the extent of species distribution. So the most narrow distribution we consider the category 1 is a species only found in Indochina, and the largest extent of distribution would be paleotropical found in Africa. Then we use the data to calculate the indices of diversity and evenness. Then we measure the environmental factor, including the rainfall available of nearby stream, and we use the presence of species in specific habitat in correspondence analysis to see habitat preference. We recorded 368 species from all 25 subfamilies known in Vietnam currently. That's almost as many as species in Australia. The dominant is Nymphalidae. We found some species is new to Vietnam and some is new to Central Highland. Section 1 and 2 are lower area, and section 3 and 4 are high elevation forests. You can see both numbers here. We found that higher diversity indices at lower elevation. However, we look at the composition of species that we found. Look at 2 by here, you may notice that higher elevation species tend to be from the biogeographic category smaller number, meaning that butterfly at high elevation tend to have smaller overall species distribution. So it means that it's more special for the one you find at higher elevation. We also found 9 species endemic to Vietnam. At the left corner, you can see exactly what we mean for biogeography, an 8 of 7 category. You also can see where is Central Highland of Vietnam located. Now, in correspondence analysis, we found that primary forests influence both species richness and abundance, and the dry season have stronger effects than the wet season. We look at the family Sapling. They are a strong negative relationship with the factor of season, forest type, and available upstream, where those butterflies looking for the water resources, and season and forest type are 2 principal factors. So we conclude that Central Highland Vietnam has high diversity and high endemism. We found most butterfly in Central Vietnam has an Indo-Malayan distribution, and family-specific difference in microhabitat conditions affect species occurrence, and season and forest type are 2 major factors that influence spatial ecology of butterfly. Thank you for your attention.
Welcome all of you. I'm Kalpana Mudshani from University of Kalani, Sri Lanka. Today I am going to present you about spatio-temporal pattern of vegetation distribution in a hydrologically altered tropical coastal lagoon. To begin, lagoons are highly productive areas, plus they harbor an important portion of the planet's biodiversity. On the other hand, they are very sensitive to climate variations and human activities. The problem in our scenario is that the rice cultivation in upstream areas of the lagoon has inverted the natural hydrological cycle. This process resulted in a decline in water salinities over many years. Consequently, noticeable shift of the aquatic vegetation can be identified. Nevertheless, still a very little information is available on the status and spatio-temporal variation of the lagoon after the change. Therefore, our objective is to study the aquatic community composition and the structure, which is known to be good indicators of an environmental change. This is the study location. The Umbilical Lagoon, which is indicated by the red square, is the affected lagoon. It's situated in the Boondala National Park, which is the first Ramsar wetland in Sri Lanka. This lagoon supported rich migratory bird diversity in the past. Initially, this lagoon was a shallow brackish water lagoon with a vegetation of cattails and sedges. In the methodology, we divided the lagoon into 3 sectors, and the plants were categorized into 4 groups according to their habitat. A coordinate grid system was used to study the plant sampling, and in each grid, sampling location, aquatic vegetation type, major species and minor species were recorded according to the cover. These are the major findings. The aquatic vegetation was algal or macrophytic, and the vegetation cover is polyspecific. 20 aquatic plant species belonging to 18 families were identified. Totally 5 submerged species, 7 emergent species, 5 free-floating species, and 3 plant species attached to substratum and has rotting leaves were identified. This diagram shows how plant species distribution in different sections. Umbilical northern section has 16 species, while middle section has 11 species along with 10 species in southern section. Moreover, the inlet of the lagoon is located in the northern section and the outlet is situated in the southern section. Number of species reduces from inlet to outlet. These images shows the most abundant species in the Umbilical Lagoon. Iconia crassips dominated in the northern section of the lagoon, while Nymphia stellate covers the middle section of the lagoon. Protamedron crispus dominated in the southern section of the lagoon. Typha angustifolia covers all the finches of the lagoon, while Cara vulgaris covers the benthic region of the lagoon. Another interesting finding was that there is a seasonal variation in the vegetation distribution. The main difference is the Typha angustifolia plant cover and Nymphia stellate distribution. Both species has higher distribution during the dry season than the wet season. You can clearly see that variation in these satellite images also. In conclusion, there is a seasonal vegetation changes in Umbilical Lagoon, which is a short-term temporal fluctuation due to hydrological dynamics. Most of the species identified in the Umbilical Lagoon are aquatic weeds and predominantly freshwater species. In contrast, Csas indicate that the current salinity regime in the Umbilical Lagoon is optimal for the distribution of freshwater plants. Thank you.
My name is Sean, and I'm here today to present to you the clustering of spatially associated species, a methodological approach towards understanding patterns in species distribution. With the ongoing biodiversity crisis, there's an ever-increasing need to evaluate species vulnerabilities, the impacts of threats like deforestation and climate change, and models to help better inform conservation decisions. However, with so many species being implicated, we also need a way to properly consolidate and summarize huge amounts of biodiversity data. Considering the spatial context of these analyses, it makes sense for us to classify species into spatially relevant biodiversity units, which requires us to understand and identify patterns in species distributions. The importance of such groupings is not unrecognized. Studies often focus on species groups with associations like montane, coastal, or lowland. And this is because we understand that species with certain distributional patterns are more or less affected by different threats. However, such groups are often heuristic or qualitatively formed. They might not be based on species-specific spatial associations. They might not capitalize on the recent advancements in species distribution modeling or the greater availability of biodiversity data. As such, for this study, we seek to develop and illustrate a methodological approach to cluster spatially associated species, and also to demonstrate some of the unique analyses that are made possible through these clusters. The clustering of spatially associated species can be divided into 4 steps. First, obtaining species distributions, which can be IUCN range maps or outputs from SDMs. Here, we use the modeled distribution of 385 tree species in Borneo. Second, to produce a dissimilarity matrix, we calculated interspecific spatial associations across our target species. We used the indices listed in a recent study by Kyle et al. 2021, which had also evaluated their performance. This included indices like Spearman's correlation, Hellinger's distance, and so on. Next, we evaluated the performance of 7 different clustering algorithms. While faithfulness to the original data was best accomplished by the average algorithm, Ward's algorithm had produced stronger and more balanced clusterings. Thus, for each dissimilarity matrix, we developed candidate hierarchical clusters using these 2 algorithms. Finally, we implemented stopping rules to determine the optimal number of clusters. The L method by Salvador and Chan, 2004, aims to identify the elbow of a given evaluation plot, which we applied for merging height and between cluster variance. We also implemented our own test of within cluster variance for each bifurcation or partitioning. We then combined evaluations of the dissimilarity indices and dendrogram performance, and also the level of variation between stopping rules. We found Hellinger's distance in combination with Watt's clustering algorithm to perform the best, and selected the optimal number of clusters according to the bifurcation paired t-test. There are many applications of the CSAS approach. We can examine the dendrogram itself to understand topological relationships between clusters, like the early separation of clusters 7 to 10 and clusters 1 and 2, which highlights how distinct their distributions actually are. We can also analyze how the structure as a whole may have changed over time due to the introduction of an invasive species or climate change. We can also construct our spatially relevant biodiversity units by stacking and calculating the relative species richness of each cluster. And this gives us a better perspective on the topological relationships previously mentioned. You can see how cluster 1 and 2 have a distinctly more western distribution, with cluster 1 species being mostly distributed along the coastlines and cluster 2 being distributed more inland instead. And these biodiversity units can also be used for further analyses, like the loss of biodiversity due to land cover change. Using the ESA land cover maps, we can calculate the proportional loss of biodiversity for each cluster. Immediately, we can see two especially vulnerable groups, clusters 1 and 6, which have a more coastal, swamp, or peatland distribution, where we know a majority of which have been destroyed to make way for oil palm plantation. On the other hand, clusters 7 to 9 suffered minimal losses, and this is likely due to their distribution at elevations that are more difficult for human development. It doesn't have to stop here. These biodiversity units can also be used for conservation prioritization and identifying potential gaps in biodiversity data or assessing trends in the evolution of species niches. Such results would not have been possible with maps of total species richness or across species averages of biodiversity loss. In summary, what I present here is not only a statistical approach for clustering species, but also opportunities for understanding the spatial structure of species communities and more nuanced analyses and summaries of species distribution data. Thank you.
Hi, hello everyone. I'm here to talk a little bit about empirical validation of computational models. The idea is that we for instance, could have a computer model that make predictions for the next 20 years. And these predictions are related to the relation between climate change and the niche displacement or extinction of animal species. For instance, we could have like animals disappearing from the earth, or we also could have like the animals changing, moving to another areas in order to find proper environmental situations and survive. But if we take the idea of validating these predictions, we will see that we don't have enough scientific works validating the predictions of the computational models, of the computer models used to be done 20 or 30 years ago. Like for instance, we could have like one scientific work published 20 years ago making predictions for our current days, but when we go to the data bank of the reports of the population of a certain sort of species, or the data bank related to the temperature of our time, you will see that many times the predictions do not correspond to the current and actual data. But even though we still have a lot of work, scientific work being published using computational forecast, and just a few scientific works trying to validate these computational forecasts. And we can see here in this graph, like for instance, since since 1997, used to have less than 200 works related to the use of the computational model to make predictions. But nowadays we have more than 1,200 works published in the idea of computational models make predictions for extinctions of animals and climate change, for instance. At the same time, we just have less than 3 works trying to validate computational models make predictions, empirically speaking. Or instead of using the keywords of validation enveloped niche models, we could use the keywords of empirical validations, species distribution models. The same idea of validate the computational predictions is here in these keywords, but you will still see that since 1996 we have less than 2 works published in the idea of empirical validation of computational models. And now, last year, for instance, 2020, we had less than 14 works published trying to validate empirically computational models. Then we have a very, very distance between the use of computer models and the empirical validation of the computer models. And in this presentation, I will talk about this problem and which sort of better assessment we could be doing to improve this situation.
Hi, hope we all in a good and healthy condition. So I'm Yanuar from Indonesia, would like to share our research project. First of all, I would like to thank the ATBC team who held this excellent event, and also I want to thank to all attendees who joined this event. I'm so pleased to be able to share our research project about the fragile limestone island forest characteristic in Gam Island Raja Ampat. There is the biggest limestone island in Raja Ampat, named Gam Island. This island has known for its beautiful archipelago, beaches, corals, and forests, not only for travelers but also for researchers. Although this beautiful ecosystem is one of the best attractions, at the same time this area is so vulnerable when its forest cover changes. This study aims to know the forest characteristic in this limestone island, especially on the edge of the island. There are 6 types of habitat on Gam Island, but this research was conducted only on 3 habitats: ecotone mangrove lowland, semi-permanent swamp forest, and lowland limestone forest. From 6 sample plots with 0.25 hectare each of them, there were 121 tree species identified with a density of 247 stands per hectare. And what about the biodiversity? As we can see, it is still considered to have a high diversity by Shannon-Winner index. The number is above 4, and Evenas index has a 0.8. And there are 4 canopy structures: emergent layer, canopy, understory, and shrub layer. We can say that the area is still preserved as a natural tropical rainforest. Structure is also commonly described using the frequency distribution of stem size. As we analyze the data, lowland limestone forest has upside down J by the curve. It shows that this type has a good structure and healthy forest regeneration model. Meanwhile, on the other habitats, the curve shows that is not suitable yet. Ecotone is a transitional area that is thought to have an abundant number of species. But based on the study results, its species richness is the lowest among others. Perhaps that there are few tree species that can grow and adapt in forest areas adjacent to the sea. By the species abundant range analysis, it shows that the curves in ecotone and semi-permanent swamps have sharper lines, while in lowland the curves are gentler. This indicates that these two habitat types often undergo changes that make only a few species dominate the area. There are top 3 species with the highest abundance. These 3 species do have a high adaptation to various types of habitats in limestone substrate. If we see about the closeness of the sample area using the clustergram, the 6 sample plots can be divided into 2 large groups. There are species that are no longer found in other plots such as mangrove species. Although the number of trees stand in Gum Island are still abundant, it was still possible that some species had threatened status globally. From the total species that we found, there are 4 species are included in the IUCN Red List with vulnerable status. The threats that probably we are facing are logging and forest encroachment by the edge of island for cultivation. The results of this study hopefully can be a starting point to increase understanding and encourage sustainable development in the fragile limestone island. I think that's all. Once again, thank you so much. Have a good day.
Well, happy to be back. This was a very exciting meeting. 11 presentations, all on different topics. Very exciting. Yes, so there's now time for questions. We have about 10 minutes, 12 minutes if needed. So I was looking at the question and answer issue, and there was one question about the study on the species clustering, and the last but one, I think, yeah, the last but two, and whether this way could be used in other systems as well. And I think the answer is yes, but maybe Sean, could you say something about this?
Thank you, Johannes, for your question. Yes, so this is a very broad approach, and I think can be used for any system. Can be mangroves across the tropics, can be used for the temperate regions. And to go about doing it is to make it as accessible as possible. It's also part of our objectives. So we're trying to roll out a R package to help everyone to implement this approach to whatever system you're trying to work on. And it's also going to be used, can also be used for, it doesn't have to be SDM outputs. It can be based on your plot data, it can be based on your IUC and range maps, so long you have some sort of idea of the species distribution. Yeah, thank you.
All right, thank you very much. I don't have many questions in the question and answer issue, but I see now that in the chat that there is now quite a number and comments at least. Let me have a look. There is a positive connection to the talk of Isabel because she translated in the slides in English and in Spanish at the same time, and I also thought it was quite interesting. And the question here is, do you think a sink source dynamics for locally abundant species could explain part of what you found? That's a question to Isabel.
Thank you, Marcel, for your question. That's a very interesting question. Well, I haven't think about it, but I think, yeah, it could be that sink source dynamics. It's something that is, leading my patterns. But referring to your next question, that is that if you wonder that if the same species is called different names in different habitats or different parts of its geographical range, and how that can affect my conclusions. Well, I curate my data set, so I look for synonyms and the data set is really well curated. So I don't think that is going to affect my overall results. although maybe there is a percentage of my species that has that problem, but I don't think so because I created all the data. But I'll think about the sink source dynamics for writing the paper. Thank you for your input.
Can I have an additional question to this same issue that I wrote here, and that is, you show that many of these patterns are possible, and so it's not one pattern, but in fact all of them are available. My question to you is, are there a few ones that are more abundant than others? I guess so.
Yeah, I mean, you mean like at the, can you clarify your question? So,
I mean, you had three of these hypotheses that you mentioned, and none of them were correct. And so then you said, well, there is some relations are positive, some are negative. That means that there is a whole variety of patterns that are possible.
yes, correct.
And still, not all patterns should be the same, and so some should be more abundant than others. Do you? And you have the value for every species, I guess.
Yeah. You're right. I mean, the different hypotheses that we test, no, we don't have proof for all of them, right? So it seems that the species come in my area. So there are rare species in terms of the geographic range size, but that can be very abundant. So that's why there is the negative correlation between okay. That, right? And yeah, I mean, there are other species that can be rare in different ways, also in a combination of ways. So, but that was the hypothesis of Rabinovich, right? That she said, like, there is these three axes of rarity, and each species can combine this in different ways, so we don't have any relationship between them. But in fact, I found relationships, right? So meaning by that there is a relationship between those axes, but not the ones that we were testing. Yeah.
Okay, thank you very much. Mm-hmm. Then I have another question here for Tirukumaran. Someone said, Marshall, I think, very cool talk. Why do you think tree richness is higher than sapling seedling richness? And I had the same question here noted down. So there's many trees, few saplings, and even fewer seedlings. Why is that?
Because well, thank you for your question. Because in that community forest, there will be because people grow the tree, and during a special season, there is some seedlings are available, and the trees are the people maintain the trees. That's why the trees are higher diversity, and the San and Vena index also indicate that the species, tree species, is higher than sapling and the seedlings.
Yeah, of course, that's what you show. But I mean, the question is, why is that? And so because all the big trees start as small trees. So one way or another, in the past, the diversity was a lot higher. And then the question is, why is the diversity at the moment much lower than it was in the past? I think that's the question that I translate here a bit. So is that, for instance, that there is more trampling or cutting or more disturbance recently?
Yeah, cutting, and the season is one of the main reasons, because the trees because they survive all the seasons, but the seedlings should have to grow due to the season is main reason for that.
Okay, the season. So you measured in one of the seasons and not in the other one, and then it's going. Okay, yeah. All right. Then there's another question for Kalpana. Really interesting work. It's impressive that freshwater plants can survive in the brackish water of the lagoon, let alone the dominant plant. And the question is, why is that? So why the freshwater plants do so well in a brackish lagoon? And some of the plants we even know here from the Netherlands, like Tifa. Angustifolia. We don't hear you. Yeah, I see that your microphone is not muted. So maybe mute again and then open again. No, it's not working. Try again. If not, we can come back to this later, and then please look at that. And if it's impossible to connect, then maybe you can answer in the chat. Then we go to the next question. Very interesting talk. Januar, why do you think one of the lowland sites were more related to the ecotone? I'm not sure I understand the full question, but that's maybe you do.
Yeah, I can try to answer the question. Yeah, based on the topographic on the island, that's the coast is straight forward direct to the lowland forest typical habitat or ecosystem. So yeah, the typical some of them we see or we're finding the mangrove ecosystem and then swamp forest and then lowland, but some of them like mangrove and then strike forward to the lowland forest. So that's kind of interesting forest type in this island.
Yeah, it just on a small area there are several habitats available. Okay.
Yeah.
Yeah, there is another question for you, and that is from Johannes, and that is about the main threats of the limestone forest on the island. And so, is that logging or is that agriculture, plantations? What is the main threat on the forest?
So it's more like the culture that community still doing like traditional slash and burn farming in here. So like nomaden farming like that. But I think it's not big enough, but it's still continue from the past to present. So it's like big deal to concern about it.
So it's more agriculture. Okay.
Yeah.
All right. Thank you. I have another question for Megan from Elliot. And the question is whether you could give us an idea of the logging intensity in your plots. Is that high? Is that percentage of stems or basal area, etc.? Because you said there is low intensity. That's what you were evaluating, but not how high.
Yeah, so a previous study measured logging intensity in the concession, and it's at 0.82 trees per hectare squared, which is really low. And then Elliot asked a follow-up question, and he was saying the number is really low even for Costa Rica, which is very conservative, and he said maybe due to bigger stems. Gabon is really interesting in the context of its forests. So Gabon is the second most forested country in the world and has 60 to 70% forest cover. And so they have a lot of forest. And I think that's why the logging intensity is so low, because they have a lot of forest to log. I was able to look up from the study. They said the DBH of the trees that they harvested was an average of 85 centimeters. So I know in the concession, they don't cut anything lower than 60. So that's sort of like a lower bound of the size of the stems. I don't know what it is in Central America, so maybe it would be interesting to look at the size of the stems and how that affects logging intensity.
Well, they do have the same threshold of 60 centimeters minimum in Costa Rica, at least.
Yeah, I know bigger than that, they can have problems sometimes with the boles having the boles of the trees having holes in them, or they start to split off into branches. And so then as the trees get older, they might have more problems, which means it's less, they can get less wood from it, even though the tree is much larger. So sometimes with the older ones, they'll actually just leave them, the larger ones, because they would have to cut out a lot of the tree due to imperfections.
Well, but there is an interesting thing here is that in both cases, in both the study of Elliott and both the study of Megan, the intensity is extremely low, I would say, and in Elliott's study is less than 9% has very low effect. He says it's fine, and less than 9% is even, I think it's even bigger than the 0.82 trees per hectare in Gabon forest. In many areas in Central Africa, the density is, selection is very low, and so
Yes,
the overall effect would be not very high. But in Central America, many more trees are being cut generally, and the trees are much smaller on average. So that would maybe make a difference. I don't know. But anyway, it's a low densities that is not making very big impact then. Okay, let's have a look, Sergio, at the question for Sergio.
Time is up.
Time is up. Wow, time is up. Okay, then there is possibilities to answer questions directly in MOA. That's also MOVA. That's possible, and you can keep on connecting. I would like to thank all the presenters for their interesting talks, and all the people that assisted, and all the people there. And thank you, Javi, for your support. And see you later this afternoon. Enjoy the rest of the day.
Thank you.